In the inactive form of the sonic hedgehog signaling pathway, there's two membrane proteins that reside inside the cell wall, patched and smoothened. There's also a protein domain, SUFU, that inhibits proteins GLI2 and GLI3. In the active pathway, sonic hedgehog binds to the patched membrane protein and inhibits it. This releases the inhibition that patched had on smoothened. Smoothened then works to inhibit SUFU and release GLI proteins. The now released proteins are able to make their way into the nucleus of the cell. In the nucleus, they bind to sonic hedgehog target genes and begin gene expression. A single plant, the corn lily, can have detrimental effects on the developing embryo. The roots of the corn lily contain cyclopamine, a teratogen, and when farm animals consumed the lily, they gave birth to unusual animals that appeared to be real-life cyclops. Cyclopamine unfortunately has a very high affinity to the smoothened membrane protein. It binds to it and prevents it from activation. With smoothened inhibited by cyclopamine, it can never work to inhibit SUFU. Therefore, the GLI proteins never get released into the nucleus. Sonic hedgehog plays an important part in the brain of an embryo. The neural tube contains developing parts of the brain, known as the forebrain, midbrain, and the hindbrain. The forebrain undergoes 3 different cleavages: horizontal, transverse, and sagittal. The first cleavage creates the precursors to the eyes, which become 2 eye vesicles. PAX6 is just a singular protein necessary for the eye development, where the single eye area will be divided in 2 and move to opposite sides of the developing embryo. MID1 and GLI3 are also needed, as MID1 degrades PAX6 and GLI3 works together with PAX6 to develop the eyes further. PAX6 normally exists as one area, but in order to become 2 to form 2 eye vesicles, MID1 moves through, grabbing PAX6 and taking it to the proteasome to be degraded. In order for this to happen, sonic hedgehog selectively up and down regulates MID1. The second cleavage of the forebrain separates it into 2 parts, and the third cleavage separates it into the left and right hemispheres. Once both hemispheres of the brain are developed, the corpus callosum forms between them, allowing them to connect and communicate. An A-lobar brain is the result of cyclopamine interrupting the sonic hedgehog pathway. The targeted genes are never expressed, and the forebrain never undergoes the 3 cleavages necessary. Sonic hedgehog won't be able to regulate MID1 for PAX6 degradation in the eyes. The forebrain won't split into 2 parts, known as the telencephalon and the diencephalon, and as a result, the telencephalon won't exist and won't be able to divide and form 2 distinct hemispheres in the brain. Unfortunately, this embryo will develop with a singular, undivided eye, the absence of the 2 parts of the forebrain from cleavage 2, no left or right hemispheres of the brain, and no corpus callosum. Cyclopamine ingested early in gestation via corn lily roots will always result in an unviable embryo, living at most a few hours after birth. While often called cyclopia, the true name is Holoprosencephaly. This is often associated with farm animals due to their access to corn lily in fields, but it has been recorded in humans and is often the result of chromosome abnormalities. The hallmarks of A-lobar holoprosencephaly are the central eye, which is often enlarged to the excess PAX6 and no MID1 regulation, the absence of the 2 hemispheres of the brain, left and right, and no corpus callosum. With the absence of these, the brain is not able to continue to function properly and will not continue development properly. This will often lead to a spontaneous abortion. Those that do survive to birth die quickly and are presented as freaks of nature in museums for us to gawk at.